All right, we should be recording. Welcome everyone to this very first popular scientific interview that we do with an exciting scientist we've selected from the Nordic region. Hopefully, this will be the first of many. My name is Daniel and I represent Milmed Tech. We are a company that delivers sophisticated solutions to the laboratories of research, pharma, industry, and so on. We try to match needs with good equipment. But enough about me. We're here to listen to Bjørt, that we've invited, Bjørt Katrinardóttir from Karolinska Institute. And Bjørt, maybe you would like to introduce yourself.
Yeah, so hello everyone. My name is Bjørt Kragesteen, and I recently started as an assistant professor at Karolinska Institute, where I have run my own lab, and we are interested in understanding how the body keeps oxygen homeostasis using different approaches, including organoids, mice, and single-cell omics. But first, I will start with how I started out as a researcher.
Yeah, some pretty big words there, and I'm certain that we get to dive deeper into those.
Yeah.
Something that I've asked you to prepare for, because this is a prepared interview, is to give us a little bit about before all that. Where do you come from? What have you done? Because it's always so very interesting to hear about researchers and their academic journey. So this is what you have prepared for us.
Yes, yeah, I thought I will start with the beginning. So I grew up in a very small place called the Faroe Islands. That's between Iceland and Scotland. And being a very small place, basically everyone is related, I feel, or this is like how I felt as a kid, and everyone talked about who's cousins with one another. And then later I learned about genetics and continued to be very interested in genetics. So I decided to go to London to study biomedical sciences to learn more about this. And I think this, really further sparked my interest in molecular biology. And then I wanted to kind of transition a bit more towards human related. So I did a master's then in Berlin at the Charité, doing molecular medicine. And then I got the opportunity to do my PhD at the Max Planck Institute for Molecular Genetics with Stefan Muntlos and Malte Spielmann, where I really fell in love with the kind of like more like the non-coding part of our genome and how structural variants disrupt communications between promoters of genes and their regulatory elements that cause human diseases, for example, limb malformations. So here, we use like CRISPR in mice and so on. But then for my postdoc, I decided to switch gears to like in my PhD, we're very much focused at one locus at a time. So I wanted to take a more of a genome-wide view. And so I wanted to look towards human evolution and adaptation to high altitude hypoxia. So I went to Weizmann Institute and did my postdoc with Ido Amit, learning single-cell technologies because this gene regulation is very dynamic, so we need to look at the relevant cells and cell states. So here, I will talk more about this later. I discovered the molecular identity of the cells in the kidney that produce erythropoietin.
EPO.
Yeah, EPO.
Right. Yes.
That's most people know it by. And I think also these new discoveries really made this journey even more exciting to then continue to be to start my own lab here in Sweden at Karolinska Institute in Stockholm. Right.
All right. Yes, I think that's sort of overlooked that a little bit. You discovered the cells that produce EPO. That's something that most of us can understand and really appreciate the importance of. Very interesting. I'm going to let you go a little bit deeper into this research of yours as well. You have if I look at you on the Karolinska Institute website, your research group is named Molecular Mechanisms Governing Oxygen Homeostasis in Development, Evolution, and Disease. That sounds pretty wide and big to me.
Yes.
So please to us, not as initiated in the research you do, please give us an introduction.
Yes. So if we just ask, so as like we evolved on Earth and kind of unicellular organisms became multicellular, and yeah, especially then vertebrates and mammals, we to make sure that all the cells in the body get oxygen, as this is essential for all the biochemical reactions in the cells to make energy and to survive. So so my big question is, so what are the cells in the body that are critical for this, and how do they ensure that the whole body gets enough oxygen? And actually, all cells can sense hypoxia, or like the lack of oxygen, or the amount of oxygen. And this has been pioneering work by many scientists. And in 2019, the Nobel Prize was given to William Kaelin Jr. and Peter Radcliffe and Greg Semenza for their groundbreaking work on the hypoxia pathway. So basically, this pathway, or these are transcription factors called HIF transcription factors that can regulate transcription program in the cells in hypoxia. But then in normoxia, when we have enough oxygen, they are continuously degraded by enzymes that use the molecular oxygen to target these transcription factors for degradation. So this is at kind of intracellular level. But then we have organs and tissues and systems in the body. And one of the key systems out there, or like in our body, that is important for oxygen homeostasis is erythropoiesis, or red blood cell production. So every second we produce more than 2 or 3 million erythrocytes, or red blood cells, in the bone marrow. And this process is critically relies on the hormone erythropoietin. And this hormone, or EPO, is produced in the kidney. So there is this interplay between these two tissues, but the blood goes everywhere in the body, so in this sense provides oxygen. So this process needs to be regulated very well. And one of the very interesting evolutionary aspects of this is that Tibetans that live in high altitude hypoxia, so around 4,000, 4,500 meters altitude, where you actually have a reduction of about almost half of the available oxygen. So the low pressure reduces the amount of oxygen that goes into the lungs. And this is really a severe challenge for our bodies. Perhaps some of you have tried to hike in high altitude. I have tried that, and I could certainly feel it on my breath. But basically, Tibetans, they have lived for more than 1,000 generations in this environment and have undergone genetic changes that have actually changed the regulation of their red blood cell production to make them more fit to survive this environment. So basically, I look towards Tibetans as an inspiration. They give us some clues where we should look for like the critical genetic elements and the critical cells that are central for systemic oxygen homeostasis. And one of the protections mechanisms in Tibetans is that they actually have a modulation of this production, so they don't produce too many red blood cells in high altitude. So if you or I will go to the Himalayas, our body will start to generate more EPO because we have some hypoxia in the body, and then therefore have more red blood cells in the body. But that also might be helpful in the beginning, but it can make the blood too thick, so it starts to clot together, and you get high blood pressure. And this is also very problematic in pregnancy. But basically, Tibetans are very much protected from that. So this is for me a source of inspiration, like to learn from them.
All right. So this is very interesting to me. So we have oxygen influencing gene expression, really. And I can see where you have sort of connected this to evolution. Even though we might talk about the short perspective here, we can see where the inspiration to that perspective comes from.
Yes. Yeah, here I could also mention that so I also mentioned disease and development. So development I mentioned because, for example, in development, the liver is responsible for producing this hormone, and actually erythropoiesis is also in the liver, and then it switches to the kidney. So yeah, it's also understanding. So how does the body to decide this switching, and why kidney in the first place? Like it does a lot of other things as well.
Right. So
And
I
then if
see
you,
I see why the perspective has widened to cover all of these things. What happens
yeah,
in normal situations, what happens in disease, and what can that teach us about the normal situation? Okay.
exactly.
Uh,
Yes. And
what a
also
privilege.
about disease. So if you disrupt the kidney function, you also start to disrupt EPO production. And this happens, for example, in severe, like in chronic kidney disease in later stages. So then you need to inject like either recombinant EPO that also some as sports people like to inject as well to enhance their performance, or yeah. And now there are also some new inhibitors on the market that are targeting this HIF pathway that I mentioned here as well. So
Well,
there
I
are some
did.
exciting developments.
Yes. So this all ties into, I would say, the question I've asked you beforehand, if there's any scientific achievement that you're especially proud of and that we should look a little bit closer at. And I think all of us have started to understand that it's related to EPO somehow. And you said that you discovered the cells, correct?
Yes, so I would not say I was for sure not the first one to describe them. So actually it's been known since the 50s that the kidney is responsible for EPO production. So this was actually by a Danish scientist where he like removed the kidneys from, I believe it was from rabbits or from rats, and could help. They became anemic basically, and then people have been trying to look for the cellular source of EPO, and this has been quite challenging because, first of all, most of the cells in the kidney are these tubular cells of the nephron that are filtering the blood. Yeah, and these EPO-producing cells, they are sitting between these tubuli, like in the stromal compartment, and so there it's they are much more rare in the kidney. And then another aspect that has complicated it is that you need hypoxia to activate the EPO gene or the transcript. So you also need to have this hypoxia stimulus. So, for example, to get human hypoxic kidneys is not that easy, but you could, for example, put mice in hypoxia. So this is something I have done in my postdoc. Um, but even then you need to somehow catch these cells. And I think this has been the major challenge, that there are no markers. People then have used transgenic reporter mice, and this has helped a lot. So this is what really helped me in my study that I will tell you about. Yeah. And also what I wanted to mention here is that even if you put, for example, a mouse in hypoxia, you have a burst of EPO transcripts, but then even in continuous hypoxia, it gets downregulated again. So you also need to know like when to catch the cells.
You have a narrow time window there to catch them.
And you also have kind of like an on-off state. So not all the EPO capable cells produce EPO at a given time. It's like very localized, depending on the localized tissue hypoxia that the tubular cells are actually generating. So it's a very interesting kind of cellular architecture that is going on there. And yeah. So what I did in my postdoc to kind of overcome this challenge is to collaborate with Roland Wenger at the University of Zurich. They had this EPO transgenic reporter mice, where basically when you give them a drug for 5 days, then any cell that produces EPO will be labeled red, or like with TD tomato protein. And then you can also give them a burst of hypoxia for 4 hours, as I mentioned, or just have a control of normoxia. So we would actually have red cells in both of the kidneys of these mice, both control and hypoxia. And then I took these kidneys and I dissociated them using enzymes and mechanical dissociation. So I get single cells. And then you can use advanced machines like flow cytometer that can excite this fluorescence of this protein, and you can sort these cells specifically that are red. And then with the innovation of single cell technologies that Ido Amit, where I did my postdoc with, he developed this plate-based single cell RNA sequencing method called MARSeq. I could then sort the cells, one single cell in each well in a plate. And then this plate is then barcoded, and you can, so each cell gets a unique barcode. It's a bit like when you go food shopping, every item has a barcode. So you can buy everything together, but you will always know what each item is. So in this sense, you also know the identity of each cell. So you can catch also the mRNA of these cells but sequence thousands of cells in parallel. So this is what I did. And you can show the next slide. So, and I created this kind of EPO-enriched kidney atlas from these mouse kidneys. And I could finally catch these cells. So this was the first time anyone could ever, really catch the cells at a single cell level and define their molecular identity. So here I discovered, so we see here, this is just a plot. So the red blob or cluster is this EPO cluster. And on your right, you will see this gray version of this map and just where this gene is expressed in red or yellow and purple. So in a subset of this cluster, we see this EPO-producing cells. But if we look across this cluster, we see it's still the same cell type, but just without EPO transcript. So if we look at other markers of these cells that we discovered, we see that they are quite uniform across the cells, whether you switch on EPO or not. And we saw also that they are fibroblast-like cell type, but different from classical fibroblasts and different from parasites, for example, renin-producing parasites. So it's really exciting. But then the question was like, are they really different from these other stromal cell types, or is it just a boring fibroblast that now switches on EPO and hypoxia? So we did some expression comparison or differential expression analysis between these EPO cells and fibroblasts. You can show the next animation, and parasites. And we saw that there were like hundreds of genes that were differentially regulated, showing that there were different cell types and not just a cell state, as is discussed in the field. And on the third plot on the right, we plotted the EPO cells from namoxic experiments and hypoxic experiments. And could see that, we see some differences, but this seems to be more of a hypoxia transcriptional program, which makes sense. And where EPO is always the strongest difference, but we also start to see some interesting transcription factors that are differentially regulated, also like potassium channel or other hypoxia factors, both known and unknown. So this was really exciting to see this cell state also present.
Okay, so it's not just EPO being produced, turned on, off and on. We're looking at something quite more complex.
Yeah, it's like a whole program. And then we were also curious about how EPO is regulated, like how come not all cells produce EPO? And it's because they have, basically specific transcription factors that bind to regulatory elements called enhancers. So this is what we can see on the left, that we see the EPO gene with this arrow, and then upstream you see 3 red peaks, and that are present only in this, now I'll come to it, that we call them Norn cells, so in this EPO cells. And these basically here, the chromatin, or like, the DNA is more open, allowing these transcription factors to bind. So these regulatory elements are only active in these EPO cells, while this is absent from the other cells. And if we look at the sequences under these peaks, both at the EPO locus, but also genome-wide, there was more than 5,000 unique such regions, which really tells me that they are more than just producing EPO. We found several transcription factors that you can show. one being like TCF21, so which was really strong, and also ZPP, GATA6. And you can show the next animation that these transcription factors are also expressed in these cells. So from this, you can show the next slide that we believe that this is a unique fibroblast-like cell type that is capable of producing EPO in hypoxia. And we named them Norn cells after Norse mythology, where the Norns are these three wise women that weave the destiny of all beings. So deciding who lives and dies.
So you got to name these yourself?
Yes. Yeah.
Yes. Okay.
We suggested that in the manuscript, and we were hoping that the reviewer would not kill us over it.
Okay. Apparently they did not.
Yes.
Okay.
So,
all right. Quite fascinating. Not just as simple as finding a simple cell producing a simple protein. Then we're talking about a very complex molecular machinery.
yes.
And I can understand then why your purview is so wide as it is, and why you're continue to go into this. Thank you for that introduction to your greatest academic achievement so far. I've prepared another question for you, because of course, as we all know, scientists never work alone, right? So today you have a research group at the Karolinska Institute, and we would like to hear more about them as well, if possible.
Yes. So when I worked on these Norn cells from mice, and we also worked on human kidneys, but it was really challenging to work on human kidneys, especially to induce hypoxia. So I really wanted to then have an in vitro model system of these Norn cells. And there are many kidney organoid models out there. So one would say that they are kind of a developmental stage of human kidney development, like a 3D model, like a mini kidney. But when I looked at the data, the Norn cells don't really seem to be present. So often these organoids are immature. So either the cells are not mature enough. So I thought that if we take some of these transcription factors that I identified in my study and switch them on during this kidney organoid development, we can help to reprogram them to become Norn cells. So this is something that Daisy and Matteo are working on in my lab, to generate Norn cells and produce EPO-producing kidney organoids. So you can show that in the next animation. So currently we are trying out different protocols. And then the idea is that we have these organoids, and then we can use your very nice equipment, the Evo in vivo 300, to also induce either acute or chronic hypoxia in these organoids and see if they produce EPO. So that's the idea.
So how's it going? Do you have an in vitro model ready, or is it a lot of work left to do?
It's a lot of calibration. So now we're actually testing different organoid protocols. So I think next week we will have the first kind of ones ready.
We should have had this interview a week later then, I guess.
Yeah, or again at a later time point.
Certainly.
Yeah. So it's very exciting to have started this work in actuality. But we also have other projects going on in the lab, also using human tissues, where we're trying to do some ex vivo culture slices of kidney tissues and other tissues and see if we can induce temporary hypoxia. And then we also use mice that we put in hypoxia and use also like spatial transcriptomics. And this is something that Katarina is driving in the lab. But we're also growing, and I'm also looking for postdocs and students to join the lab on the mission to decipher the identity of the key cells in the body and the regulatory mechanisms that underlie oxygen homeostasis.
Okay,
And
hopefully we can find some more applicants then through this interview. That would be fantastic. I'm a little bit curious, if you allow me to ask unrelated questions perhaps, but when you study these human tissue samples, from where does those human tissue samples come from? Is it deceased patients, or is it?
yeah, so mainly in disease. So for example,
Yeah.
the kidney samples mainly come from patients that do or undergo partial or radical nephrectomy, so taking out a part of or the whole kidney.
Mm.
And often they have a renal cancer, like renal cell carcinoma or something. So, and then we get the adjacent non-cancer tissue. But the
Okay,
question,
okay.
like that was also a challenge, like how healthy is that kidney? So there are definitely some ethical challenges. So so this kind of in parallel to the organoid system, and also using mice, like yeah, you have to work with what you like ethically can do. Yes,
Yes,
so
because of course you want to start with an in vitro model, but sooner or later you need to look. This is a challenge, I think, for many researchers out there, not just yourself. You have some grants here you've also
yes,
promoted. Should we mention them?
yes,
Would that be polite?
yes. Yeah, so basically, the reason why I started here at Karolinska Institute, so I applied for a position in the Department of Medical Biochemistry and Biophysics, and they have these every year now kind of frontier grants that you can apply for, and then you compete like internationally with many other excellent people. So I was very excited to be offered this position, so I got a starting grant from them. And then I also applied for starting grants from Vetenskapsrådet, like the Swedish Research Council, and the Swedish Medical Research Council, where I also got starting grants. So we can, do all the experiments that we want to do, at least for now. And then hopefully some of it will work out, so we can continue our research.
The struggle that
Yeah.
every PI faces, but you're a PI with your own research group. You've ended up at the Karolinska Institute as a PI with your own research group. Something that I'm very curious about is this a calling to become a researcher at this depth, or is it a career choice that's very selective, or a duty to society? How do you feel about your own role as a researcher in molecular and cellular biology?
Yeah, I'm not sure if I have a straight answer to that, but I would say, yeah, it's curiosity driven. Like I feel it's quite a privilege to try out the ideas that I come up with, and also in discussion with many other people. I think most of the ideas have developed in conversation with other people or reading other people's work. And then like a really fantastic environment with international colleagues, and to teach students and to mentor, and also to develop as both as a manager and as a leader. I think it's really exciting. And and I think also somewhere this commitment to conduct ethical research and also sustainable research, and also I would say kind of a sustainable research culture. So that's really important to me. So I
So
feel like
a
I
little bit
started.
of everything,
Yeah,
really.
I feel like I
Yeah.
started already doing experiments when I was a child, with I don't know, dead birds or that my cat caught.
All right.
So I think it's curious about how things work in our body, and I feel privileged to be able to do that on my everyday basis. Yeah.
Yeah. And it's a privilege for us to take part of that answer. It's never quite as simple as we'd like to things for things to be, right? Of course, you and I, we know each other because you've bought a machine from us, really, the Vivo 300 from Baker. And for those who don't know what this is really a cell culture workstation. You can do cell culture and cell manipulation in a precisely controlled environment. A big focus here is, of course, the O2. But when you think about O2 and cell culture, you also need to think about CO2, temperature, humidity, and so on. So think about it as an advanced incubator with oxygen control. You bought this from us. How is this going to help you dive deeper into your research, do you think?
So I think this will help me in several ways. So both we can use it to produce more like physioxic conditions for the cell culture part. So I mean, when like during human development or any development, then there's not 21% oxygen present. Like it's much lower, and it has been shown that, reducing the oxygen concentration actually helps to develop or like to grow, for example, stem cells. They need a hypoxic environment. So that's one way like that we can play around with these organoid protocols and oxygen concentrations. But then also in the mature organoids that we can induce either acute or chronic hypoxia and study the molecular mechanisms and dynamics in gene regulation during this hypoxia stress.
All right. Is it important for you to be able to simulate hypoxic stress quickly?
Yes. Yeah,
Then
so we will also do very short, some short time courses also.
Yeah, then I might have a suggestion, but we can take that after the interview.
Okay.
But we have a method around that. But thank you for letting us know how the things we sell and service and maintain, what they do and what they help people do. It's always wonderful to hear that. And as a chemist, I have to apologize for not making lowercase O2 and CO2 there. I just realized, but that's how it goes. All right, we're gonna sort of slowly end this interview together with you, Bjørt. We have used some images from the public domain on the internet, and those are credited here, the authors of them. And following that, I would just like to say thank you for joining me for this chat. It's been very, very fun for me, and I hope that you've enjoyed it as well.
Yeah, thank you for inviting me, for the opportunity.
Yeah, we look forward to working together moving forward. Right then, I'm gonna end this recording.